Hi, Chaitra and Manvita. I hope I'm audible and clear.
Yes,
Yes,
ma'am.
ma'am.
Okay. So we'll start today's session. So 1 second, just let me share my screen. Okay, so this is session 4 for metagenomics. So in this session, we will be studying we'll be doing the continuation part. In the last session, we have studied about the KIME2 installation and the Conda activation, and we have successfully installed as well on our systems. So it's just a continuation part for the day where I'll be talking a little more about KIME2 installation and environment setup. And then we will try to understand the Conda environment and the KIME command lines and how it handles the data and everything. So, like before we do any microbiome analysis, we should ensure that our system is like properly configured. Otherwise, even if there's a small error in the setup or in a like pipeline, it can harm the entire process of us visualizing the data. So that is why we need to understand each and every step, and each and every step should be correct so that we can move on to the next step without any particular error. So as we have like very nicely installed entire KIME and Conda and everything, and you guys are a little familiar with that as well. So now we'll move on. So what and all I'll be covering for today's session. It will be first we'll understand the Conda environment management and how are we going to manage this KIME and Conda environment. And then we'll learn the structure of KIME2 command lines. So when we use KIME2, so we use a commands. That is, we directly write commands in terminal. That is what we called as command line interface, which I have taught you guys before as well. So we'll be learning about how is this KIME2's command line system works. So after that, KIME generates two different files. One is a .qza file and one is a .qzv file. .qza is an artifact file, whereas .qzv is a visualization file. So we'll be understanding about both the files as well and how to analyze that. And then we'll prepare the system for microbiome data analysis workflow. That is, we'll be checking for all the plugins, all the dependencies are there present, so that we can run our pipeline without any problems, and we can move on to the next steps. And then I'll be also talking about certain common errors and troubleshooting strategies to overcome those so that we don't face much errors in the latter part of the process where we have to run our pipeline properly. So these are the foundations. Without these, you cannot proceed in a microbiome analysis, and we'll be talking about this in much more detail. So now first question is that why is an environment setup critical? So like why are we setting up an environment in Conda? Why not working directly there? So there are multiple reasons for that. So as I told you, like let's consider Conda as an entire computer system. So, or you can say Conda as an entire building. So in a building, there are multiple rooms, right? In a multiple rooms, different families live according to their own comfort. So that is how we will define an environment here. So an environment is like one room in an entire building. So Conda is an entire building where inside that there's a small room that is called as environment. So that environment can be as of your choice. Whatever you want, you can install there, whatever you need, whatever dependencies. Like it is however you want to live, you can live in your entire house. So nobody can come and interfere with you, and nobody can question you, why are you doing that? So similar way, you can have your own zone, you can have your own environment for specific tools, and then you can work in that. So why is this environment setup so critical? So first is dependencies and mismatches. So when we use bioinformatic tools, so bioinformatic tools largely depend on multiple libraries. They don't work alone. Rather, they work in as a multiple libraries. So what happens is these multiple libraries, their versions can be different for each tool to work. So say for one tool, you require a version 2.1, but for another tool, you might require a version 2.2. But installing that into your base environment may cause some critical problems. That is, you won't be able to run the tool that requires a different version which is not installed on your system. Then you might need to delete it, reinstall it, but then it will become like you can only use one tool. But we don't want that. We might need both the tools in the same pipeline, and if this version mismatches happen, they can break the pipelines. So it will be a little difficult for us because our pipeline will keep on failing because there are version mismatches in the entire protocol that we are using. So this is the one, like this is one of the most important problems that beginners face. So I also faced this problem initially when I was learning KIME, how to analyze it. This Python requirement, what Python we use is different than the version Python that KIME uses. So I had a mismatch there, and it took me a long time to understand that why is this happening? Why is this error coming? Why is my KIME2 not working? And when I realized that, then I understood this importance of this particular environment. Why do we need to create it? So similarly, so KIME2 requires a different Python version, and we can only use the Python that is compatible to our KIME2 version. If the Python version doesn't match to our KIME2 requirements, the pipeline will clash, KIME2 will not work, and you will not be able to run any commands. And like at the worst, you won't be even able to install KIME2 because the version clashes, and it creates a lot of problems. So what are the benefits of using this Conda? So Conda is basically like it will create a separate room inside your computer. That is, it is going to isolate that particular environment from the other things that are present in the system. So it will give isolation, it will give stability, and it will give you reproducibility. So if I have to explain this, that is, you can stay in your room. The analogy that I just took, you can stay in your room in your own zone, in your own way. You can buy whatever you want. Like you don't have to share it with others, and that is going to be your specific. So similar benefits Conda also shares with us, and similar benefits. One second. Similar benefit Conda also shares with us, that is, it provides isolation, it provides stability and reproducibility. So you can just work in a separate environment where you don't have to worry about the tools and the dependencies, because whatever you require for that particular tool to work, you can just install it in that environment, and then you can do the entire protocol and workflow, and you can run your pipeline accordingly. So this is good because it does not clash with the other dependencies that are required by other tools, and you can have a separate environment for all of them. Like you can create multiple different environments how much ever you want, and then you can download the desirable packages in each of the environment, and you can work very easily without any problems, and no clashing will happen, no mixing of the tools will happen. You can just switch the environments, you can go to other one, you can do your work, and then you can come back to the previous environment whatever you want. So like it becomes easy for handling, it becomes a little manageable, and in a like in a proper you can say in a proper functional manner, that is no mixing and match of anything, everything is a separate entity. So what is a Conda environment? I think I've already explained this. That is basically it's a self-contained software environment. That is a Conda environment is a one particular environment in the entire system where you can install all your dependencies. So as I mentioned before, it is like a separate room inside your computer that you're giving to one particular tool where you can install everything that is required for that tool to function. So like for this particular, there's one environment, Conda environment, that is shown here. So it has its own Python version. Then it has its own libraries that is required for the tool that we're going to run in this. So NumPy and SciPy are just some Python libraries that we use for data analysis. And then we have some tool specific dependencies. So whatever the tool requires more for us to install, we can install that in that environment and work freely. So it is a beneficial thing because it prevents any conflicts that can happen between two tools, and it can allow multiple environments on the same system. So if you want to work in multiple environments, say you need to do KIME. After KIME, you need to do certain alignment part. So you can install an alignment tool in a different environment. So you want to use HiSat tool. So you install HiSat 2 in a different environment. You download KIME2 in a different environment, and like this you can have multiple different environments where you can have different set of tools. You just activate one environment, use the tool, deactivate it, and you can come back. So you can always shift from one tool to another, and you can go back forth or everything, and you can just analyze whatever you want, and you can get your results and work accordingly. So next is the KIME2 environment structure. So how does it So basically, for us, whatever, like you guys can confirm, right? The environment that you guys created for KIME2, it's the same name, right? KIME2 Amplicon 2024.10.
Yes, ma'am.
Yeah. So we'll work with the same environment name. So this we have installed. And now this environment, last time when I asked you to check the version and for the help section, you got multiple plugin tools, that whatever tools that this KIME has already inbuilt. So these tools are the ones that we will be using further for analysis. So we know that this KIME2 has KIME2 core tools, that is whatever is required, like import and visualize all of that. Then plugins involve these data diversity data too, which we learned for denoising. Then diversity feature table to generate a feature table. So these are all the Python. So these are all the plugins that are already contained in an environment. And then we have the Python dependencies. So wherever Python, whichever version we required, we have that installed with the KIME2. That's why our KIME2 environment is working very nicely, so that we can work it further with. So this is just a pictorial depiction of what I just told. It is KIME2 core tools. What are the plugins, and then the Python dependencies. So all of this, like it's like entering the like correct laboratory and performing an experiment, like before performing an experiment. So basically you need to have everything set for performing an experiment. So you need to have everything already predefined, and whatever is required should already be set up, so that you know what you have to use and how to just put it in and use all of it. So this is how the KIME2 environment structure work like. And then we can just use these plugins that we have installed along with that, and we'll move on to the step of analyzing it using a command. So next is activating the environment. So activating the KIME2 Amplicon, this is the command that we can use to activate. And then what will it do? It will basically change the active environment, and it will update the path variable, and it will enable the KIME2 commands. So if you do KIME2 in the base environment, it will not function because KIME2 is not installed in the base. Rather, KIME2 is installed in a particular environment named as KIME2 Amplicon. So I'll just show you guys once. So let's say this is a base, okay? So now we'll do conda environment list. So it will list all the environments that are present. I've already installed base environment. Base is the main one. And then I've already only installed the KIME2 Amplicon. So what I'll do is I will copy the entire name. Then I'll do conda. So for doing that, I would like to show you guys here. So if I do KIME health, it will not work because it will say that KIME doesn't exist because the KIME command is not found in the base. But if I activate the environment, then I will move on to the environment where I installed KIME. I didn't install KIME in the base, so my KIME commands will not work in the base environment. But when I will move on to my particular environment, that is the KIME Amplicon environment, and here I'll do KIME help. Here it should work, and it should show an entire table of all the plugins, of all the commands that it has installed. So that is why I'm saying that this environment is a separate environment. This environment will not get mixed up with the base or any other environment you create. So this will only remain in this particular environment where you have installed it. So now this is how we check the help. So basically you don't need to memorize all the commands. There are commands and there are tools and how to use that. I will tell you the structure of the command, how a KIME2 command looks like. But just to tell you what and all plugins have been installed in this particular thing. So I mentioned about Data2. Data2 is a plugin for sequence quality control. That's for denoising, and similarly Deblur2 as well. So both of them are used for the sequence quality control, and that we use for denoising and everything. Then demultiplexing. Demultiplexing we are using the command, the tool that will be demux to do the demultiplexing. I hope that I don't have to repeat the what demultiplexing and denoising is again, because I have already explained you guys. Are you okay with me just using the commands just using the words as demultiplexing and denoising?
Yes, ma'am.
Yes, ma'am.
Okay. And if anywhere you think that I'm using a word that you're not able to understand, you can just stop me there so that I can explain the entire thing again with a proper explanation. Then next, if we move on, then we have this emperor plot. I showed you a plot, right? That black colored plot which had red dots inside it. So that is called as an emperor plot. So this emperor plot will tell us about the, it is basically for plotting. So it is going to tell us about the closeness a species, how close a species is. Like, are they clustered together or are they distantly apart? All of that you can analyze with this. Basically, this is a plot of a PCOA. That is a principal coordinate. When I move on to that, I will explain what PCOA is and how it is working. So it will basically from multiple dimensions, it will just reduce the entire data to 2 dimensions, and then you can easily compare between that and like how much comparison is there, how much differences are there, all of that. Now, similarly, feature classifier for taxonomic classification, feature table for feature table generation, metadata is for a plugin of metadata. Basically, you have to import the metadata, right? Then in your kind to pipeline, the metadata you have is separate. Now you need to import the metadata so that you can use it to demultiplex the sequencing. So for that, you need to import. For that, we have a particular command. So all these commands are there which are present in time and that I just showed you. You can see this entire message when you do help. And if you do just KIME version, it will show you the version of KIME that has been installed. Okay, and then it is telling me that you can run KIME info for more commands, like for more information. So let's see what it shows. So installed plugins, that this and all is installed, and everything it is showing that it is which is the system. See, it is using Python version 3.8.15. It might not be same for you. It is different because the version of KIME that we're installing. So for me, the KIME version is 2023. And for you guys, it's 2024.10. So here the KIME version is different, but the tools and everything remain same because it's not that different. Sorry. And these are the same plugins that you guys also have. You can also run the same command simultaneously and just check once if these plugins match with your plugins, because we should be on the same page as we have to run the similar commands. So I'll just give you guys 2 minutes. First, you can type this command. I will put it in the chat box. One is KIME help and KIME info. You guys can just run the command once. Just run the commands and tell me. You can already see it on my screen and tell me that we guys are on the same page, right? So that, because we should all have the same things installed.
Ma'am, first we have to change the environment right now, ma'am.
Yeah. First, you have to, one of you just share your screen. Anvita, just share your screen so that I can also see what's happening. And Chaitra, you also keep following up whatever is happening on the screen.
Okay, ma'am.
Manvita, I think you just stopped presenting your screen. Just open your terminal. Anvita, is there any problem in presenting because we are not able to see your terminal in the on the screen?
Ma'am, let me try again.
Okay. Yeah, it's visible now. Hmm. First, you have to, you can copy this entire thing and you can write the Conda Amplicon, whatever is the name of your environment, and activate it.
Okay, ma'am.
Chaitra, you are doing simultaneously, right?
Yes, ma'am, I do.
Okay. So now you have activated the environment. Now you just copy paste the command, that is kime dash dash help. You can write it also if you want. Can you zoom it a little near the installed plugins? So you have all the same plugins that are installed in my system as well. It's just that they're a newer version, and that's a good thing that we will not be lagging behind on anything, and everything is same. We guys are on the same page. Are you also able to see the same thing, Chaitra?
Ma'am, can you please share that code like conda
Um,
activate kind to amplica?
okay. So you have to, first of all, you have to check the environment. So what you can do is, I'll write it in the chat box in your
Ma'am, I
terminal
checked,
first. Yeah.
ma'am, I checked the environment conda and list. I need second code like conda activate kind to amplica.
Okay, so what you have to do is you have to write conda activate, and then when you did conda environment list, you got the list of the KIME, right? Where the
Yes,
KIME entire
ma'am.
name. So
Yes, ma'am.
just copy that entire thing and write conda activate and paste the name of the environment.
Okay, ma'am.
You can share your screen as well and show so that we can confirm that you are also on the same page with us.
Yeah, sure, ma'am. Right.
Yeah, just type just type KIME space info.
What about kind help? Like, dot-
Oh, you can write that also. You can write that and check later. Right now, we'll just tally the KIME info part. You write KIME space info.
yeah, sure.
Yeah. Okay. That may you can just check. Like here, you will get the versions as well, and in that KIME help, you will like it will tell which and all plugins have been installed and how to use it and all of that. So you can later on type that command as well, and you can check. So you guys, you also have a similar version. So all the plugins are there as well. I can see all the plugins. So that's great. So the everything is happening. You can also understand, right? Just go up a little. You can see these plugins, a list of plugins that are there. Whatever
Yes,
I
ma'am.
mentioned, yeah, here. So it is data to deblur. and diversity, and then you have phylogeny. All of them, whichever I mentioned, have been been already installed. So we will be using all of them only collectively. So now
Sure.
you can stop sharing your screen. I'll present and I will move on to the next section. So I have mentioned all the commands, whatever I sent on the group already in the slide, so you can use it. So this is the Conda activate environment. So you can use this command Conda activate to change the environment, and you use the and to change the environment, it updates the path variable, and then it will enable the KIME2 commands. Because as you change the environment, it will automatically update the path of the plugins that are installed in KIME, and then after going into the KIME environment, you can directly enable the KIME2 commands there itself. So now how do we verify the environment activation? We just did all of this. So to verify it, you are you saw that, right? Your environment name appeared in the terminal as KIME2 Amplicon 2024.10. So this you got to know that this is your environment. So now you will go and activate this particular environment. And to check KIME2 installation, you will write KIME version. So after you write version, as I showed here, so when you write version, it will show you the version of the system that you are working in. So I'll just show again. I'll write KIME version. So it will show you the version of KIME. So my version is this. That means that my KIME is successfully installed on the system, and this is the version. For you guys, it will be 2024.10. So that will be the version that you guys are using. It's a more latest version. So by using this, it will be confirmed that your KIME has been installed and it is of what version. So after always an installation, you generally check if it is installed or not by generally writing either KIME version or KIME help, so that you know that your entire whatever you're trying to install the tool is properly installed. Because these commands will only show an output if the tool is properly installed. If the tool is not properly installed, they might give you an error, or they might say that this command not found, like KIME not found or something like that. So there will always be an error if it is not installed, and if it is installed properly, you can see the entire drop down menu where you can see what and all plugins are present and what and all commands are present. So now we have to manage the packages in the environment. So we should see like we should know how many packages are there and all of that. And like basically when we use this command conda list, so it will show us all the packages that are present. So say I will use this command, right? Conda list. So it's taking some time to run. So it has written all the packages that have been installed. It will be a long list. So you just don't need to work on this. Like this is not that important for us to work on with, because if our KIME is working, if we run a command in KIME and if our KIME is working, that means that all the packages that we required for KIME has already been installed. This is an additional step that you can do, where you can check once, like by running conda list, if your KIME is not working. So if it is not working, then you can check for the entire conda list, and you can see if there is any package missing, or if there is any version conflict that is happening, and then you can understand it better. And you can, this is basically for debugging, that if you have any error, if your installation is not happening proper, then you can check once conda list. Maybe this can happen that sometimes half of the package that you're installing gets installed. That means, say if you wanted to have 100 packages in a tool, it is not getting into 100, but only 50 of it got installed. Your tool will eventually not work because it does not have all the packages and dependencies. So what happens is, then we should check once that if how many packages installed, either we remove them or we reinstall everything, so that we can have the entire packages, so that our tool works properly. If you need to update these packages somehow, somewhere required, don't do it unnecessarily. But whenever the system asks that you're running a command and it is asking you that, okay, now update all the packages, or if you have been using it, like you haven't used a package for a long time, or you haven't used conda for a long time, so whenever you come and you open it, you can update it. So, but don't like, we have to use it a little carefully, because sometimes it happens that some versions mismatches happen. Like, you'd update it, the versions get updated, and that update is not technically what matching with what our kind wants. So there it might a conflict can arise, and for that problem to avoid, we should only update this particular all packages. You can update conda. Conda updation is not a problem. You can update conda, update, and all of that. That is, you should do regularly, because you should be up to date. But this you can do when it is required, like when it is absolutely necessary. You can just do use this command, and you can update it. So now exporting an environment. So when you use conda, so you should like be able to, like, it's just not about one system. If you worked in an if you worked in a particular system, now you, like, what if you shift to another system? You can't do the entire thing all along again. It will be a very long procedure. What if you have months of work? You can't repeat it again. So what this beneficially thing of conda is that it gives you reproducibility. That is, it will help you save the packages or whatever you have installed. So this is conda environment export. So here you can write what in environment, say whatever is the name of your environment, and then you can write, save it as in a YML file. That's basically a human readable text formatted file. And then you can just after exporting, now you can recreate the same environment using this command, that is conda environment create, and the same. So this environment that you had saved before, now you can recreate the entire same thing, because from the saved environment, and you can reuse it. We are not be going into much detail. This is just for an additional information, where if you guys change systems and if you want to work again and the same thing, like you need to run through the entire pipeline again, or whatever versions and packages that you have installed, and you want your work to be there, so you can use these commands and you can recreate your work in an environment, or you can recreate your entire environment so that your work remains constant. So now in kind 2, I mentioned that we will be using a command line interface. So command line interface is basically you will be typing commands on the terminal itself. So after activating the kind2 package or kind2 tool, you will move on to the environment where kind2 is installed. Now, how will you run the commands? So now on terminal itself, you will be writing commands, that is called as a command line interface. And then you will write the commands, enter, and that command will be executed and you will see a result. So now Kime has its own way of commands, like it has its own way of like, what do we say, it has the own format, like how you need to write. So there is a proper format that you need to follow. Like, you start with kind, then you write action module, the plugins, the inputs, the outputs, all of that you have to write in a sequential order, and then you have to give the paths, name of the files, all of that, and then you execute those commands in the command line itself. So in command lines, you do not require any graphical interface. You can directly write on terminal itself and you can execute. So what are the advantages of using this? It has a faster execution. The scripts can be automated, and it is suitable for larger data sets. Like, it will not hang. It might take some time. Your system should have a good processor, and it might take some time to process a very large data set, but it is generally quicker and faster, and it generally does not hang that much. So what do I mean here by this automation using scripts? So I have already explained you guys before, right? That bash script, the .sh files that we create, we can directly execute it in the terminal by writing bash and the name of the script .sh. So when we want to run the same pipeline for 100 different samples, you cannot run the same pipeline for each and every sample sitting there. What you do is you do automation. That is, you write a code. Generally, for such automation, we generally use bash scripts, because those are easier for us to, like, for the beginners, it is easier for beginners to understand and then use the entire script, because it is very similar to what commands we write on the terminal. We just have to change very few changes, and then we write it in the bash script, and then we execute them. So what we can do is we can put path for all the 50 samples that we need to run this pipeline for, and then we put it in a loop. That is, after going to one, it will go to second, third, fourth, until it reaches last. It will run this entire script for all the 50 samples. So initially, what we had to do is we had to run the entire pipeline 50 times. So much time consuming, so much hard work. A lot of time of you will go there and not analyzing the data just for running the script. So what this automation has done and why is it useful? So after automating, you just have to run one bash script, the entire command. Even you say, even if the commands that we run for one pipeline, say if there are 10 commands, you can just execute one file. You can run all those 10 commands in one go for 50 different files, and you can save your time by avoiding running commands again and again, and putting the time into visualizing the data more properly. So that is what automation is like used for. And automation using a script in kind, and that to a command line thing, it is very suitable and it is very efficiently it is very efficient. You can work very efficiently and faster because of using this. We can do automation in the end, like where we can generate an entire bash script for our kind to pipeline. But initially we will be running each and every command separately, because we need to understand what that command does, and what does that command do, and like how are we using that command, and what results are we getting from that. So this is how a kind to command structure looks like. So all the commands that we will use in kind to, it starts with the letter kind. So there's a letter kind followed by a plugin. So plugin is basically a functional module, that is the tool category. Like what you want, which tool you want to use that is present in kind, like deblur, data2, demux, phylogeny, whatever plugin that you want to say, you want to do demultiplexing. So here you will use a demux. So if you want to do denoising, and in denoising specifically, you want to do using data2. So here you will write data2. So plugin is basically the functional module that you use to basically suggest what function you want this kind command to do. So that is what you put here. Action is what action you want to do. So you want to import the data, you want to export the data, what you want to do, that is action that works on our requirement. Then parameters is the inputs and the output. So input is like what file you will give in as an input, so that like input is what file you are giving as an input, so that this KIME tool should work on that. And output is the what output are you asking. So if we are going to do demultiplexing, then we are going to give the raw files, because we want to demultiplex it, and output will be a demultiplexed QCA files, the artifact files or the visualization files. So that files we will get as an output, and the input file will be what file we needs to be processed. And parameters you can put accordingly. When we'll move further, I'll talk more about parameters. That depends on particular file and a particular port. Basically, what do we want? So this is an example command. This is a very simple command where you're importing a data. It's a sample data that you're importing. So it's just the name of the sample data. Now you write KIME tools import. So when you tools here is a plugin. So you are using a tool that is a plugin. So this is what function you want to take. So it's a very common generalized command. So you are taking it, and then you have to do import. Import is the action that you want to happen. That is, you want to import that particular data. So type is a data format. This is the sample data sequence with quality. This is the quality. This is just a random name that I've taken. And this data you are taking. So that you can import it. Now, input path. So where is this data present? The raw data file, where it is present, and the data file. So this data you will import from this particular directory. If you are running the command in the same directory, then you don't need to give a path. You can just give the name of the file, and then output will be whatever you want to generate. So here they're generating just a demux.qca. Technically, it should be like here demux. It is just a very generalized command. So you don't have to look into the technicalities right now. Just understand what each and every term is like meaning here. So what you are doing, what action, what plugin you are using, what type of data, the input data and the output data. So these are some of the parameters that are really important, and these will remain constant. There might be some parameters that keep on adding as the command as we move on to some commands, like importing the metadata, then generating a demultiplex sequence, and then generating feature table. All of that will be a separate thing. So with each command we will be learning what and all inputs happen and what and all outputs are generated. So now if you want a help in KIME, so what happens is, so you can just directly use KIME help. So when we do KIME help, it gives you the entire list of the entire commands that are present, right? But if you need a specific help, say KIME data to help. So what you can do is you can write KIME, you can write data to, and then you can do help. It will specifically show you how, what and all, like it will specifically show you all the requirements and every information about data to plugin that is present in KIME. So you can specifically open each plugin's information as well, and you can just visualize the entire information collectively using KIME help. It will provide all the commands that are available, the parameters, and how to use. So usage instructions are also present in that. So whatever is present, you can just check it on. You can see if your command, it is, sometimes it might happen that the command that you are writing shows that it is wrong. So when you see that it is wrong, then like what to do next, you can just open these help commands and you can check. So when I will be teaching each and every command, I will be using one wrong command and one correct command. So when I put the wrong command, it will show that there is some error in the command. Then we can go on to these help sections, we can check it how it works, and then we can move on to the correct command. So now I told you that there are two files that will be generated for visualization. One is an artifact file, that is a QZF file, and one is a .qzv file, that is a visualization file. So in QZA files, I told you it is generally a numerical or an informative file. It does not contain any visualization part, that is no graphs, uh, no bar charts, nothing it contains. It only contains the data part. So it will contain a data. It might contain metadata as well for certain samples, and then it will contain a provenance, that is the history. So when you work in KIME 2, there's a provenance that happens. That is, you will have an entire log of history. That is the entire log of the commands that you have used for running this KIME 2 pipeline. Because the entire it keeps this particular property of history, but I get wrong somewhere. Let's say you should have a entire entire command that you have used. Check the pipeline you went wrong and why the error is being shown. So it is good to have this provenance part and history, but these cannot be opened directly. So like you can't just open and visualize it. See, when I showed you there, the data files, either you can open it in the browser or you can view it properly separately. That I'll be teaching more about the commands, how you can use this. But majorly this file is generally for, like whenever we run KIME, lots and lots of QZA files are created for each step, whatever we do. These QZA file contain information which is generally interpreted by the QZV file. And that QZV file is the one that we visualize. So this and all is a very raw data. We will not be visualizing this much because of this, we will generate QZV files, and those QZV files are the one that we'll be focusing more on. So I'll just tell a little more about provenance tracking. So provenance basically tracks the input data, it commands used and the parameters applied. So basically whatever you are doing in KIME 2, whatever data is getting in, whatever data is getting out, whatever commands that you are using has a proper track of it. So that track is maintained, because if in later you want to say you have to go and check it back, like what is some problem is there, or some command didn't work or something like that, so then you should have a transparency that whatever command you used before, that is completely visible and that is completely shown, so that you can just go back and check what happened, like is there any command line problem, or that command didn't work, or you have to repeat anything again and everything. So when we do provenance tracking, it will help us more in reproducibility. That is, anyone can repeat your analysis as well. That's another important point. That is, if you have run an entire pipeline, if anybody else wants to, they can just do provenance tracking and run the entire command line again, whatever commands that you have run, because all of them get stored in the history. And transparency is there. No hidden steps. Everything is getting recorded. Then we have error tracking. Say if something goes wrong, you can trace back to the step and check where did the error happen and how will you be able to cure it. And if you want to publish the data, then publishing requires scientific credibility, right? That everything is noted, everything is documented. So this helps in that case where you would like to publish your data after doing a proper analysis and all of that. So provenance tracking is another important factor of using kind to pipeline, like for tracking everything that you are doing in the pipeline. So now we have the QZV files. So QZV files is the visualization file. So these files are very interactive. So like you can, I last time I showed, right, for the taxa bar plot, you can change the levels. You can see that the taxa bar plots keep on changing. You can zoom into the bar charts. So it's like interactive output. You can talk to those graphs and you can understand them. So these visualization files will include graphs, box plots, and PCOA plots. Will have quality plots, scatter plots, diversity analysis. Like a lot of plots can be seen and a lot of data can be analyzed and interpreted from these QCD files. Like you can zoom into some specific data, you can zoom into some specific criteria, you can visualize that. And like uh, the interactive, what I mean here is like there's a graph. So it is showing the entire 40 samples. So you can zoom on to the only 10 samples, or you can even zoom on to only one sample. So that is what I'm trying to you guys to make understand. Like this is what I'm trying to tell what an interactive output file looks like. So you can click on it, you can understand each plot. What does that mean? Each bar, what does it mean? Like for each box plot, also you can understand that like what and all it depicts. So it becomes much more easier to analyze and visualize the data because everything is completely marked, it is completely labeled, and it is very properly and easily understandable. And is viewing and visualization files. So when you want to view these files, you can either directly use a command line approach where you write this. So you write KIME tools view and file.qzv. File is whatever is the name of your file. So run this command directly in your terminal. It will open a particular window on the website. And on the website, you can just see it will open an HTML page where you can directly see the outputs. So you write this command on command line, will open and you can see the outputs. Another thing is when we can directly use website. So there is a time to view website where you will upload the QZV files that we will get from our commands that we run for time to pipeline, and then it will also open the same web page. So using a command line approach is very easier compared to others because you have to open a website, upload it. I will show this also. We will do with both the things, but we will be preferring more of the command line approach. So it becomes very easy. You just have to write a command. It will automatically open and you can directly visualize it and then close it and come back to the command line. So data requirements for analysis. So what and all is required? So when we basically do a KIME analysis, so FASTQ files. So FASTQ files will be the one that will have the raw data and the raw sequencing data that we have to. One second, guys. I'm sorry. So FASTQ files will be the raw sequencing files that FASTQ files will be the raw sequencing files which will have the entire data. These are the files that you will be analyzing later that by using the import command. So these are the raw data that came from the microbiome sequencing. Whatever sequence you sequenced, whatever sample you got sequenced, now those will be present in a FASTQ file. So in the FASTQ file, you can just run the commands using these FASTQ. First, we will do quality control, and then we will run the FASTQC, and then further commands demultiplexing on these files directly. Then we have the metadata. So metadata basically it will contain all the information about your analysis. Say you did a soil sampling. So in soil sampling, what and all soil, like which soil did you take, what and all samples, what time, how conditions, and all of that you can understand from the metadata. So manifest file. So manifest file is basically like we generally create a manifest.tsv file. So this manifest file will have the information, that is the path. Path is like where our files are stored. So wherever this path for the FASTQC files, the metadata file, so we have to write the entire path of the metadata files where they are stored, FASTQ files where they are stored into this manifest file. So this manifest file we are going to use for further processes. So we will not use metadata and fastQ separately. Rather, we will directly imply all our commands on manifest file, because manifest file will already have the path that where our metadata file is stored and where is our fastQ file stored. So we will be directly working on that. So we will create a manifest file when we start working on the data, and you guys will be more clearer on that fact that how we are generating it. So now whenever we work in KIME, so I would suggest using a proper directory structure so that the entire workflow and entire result section is properly stored, so that there is no confusion later. So you can create a folder named project. And this is just a sample directory structure that I'm showing, so that the workflow is organized and it is easily navigated, so that you can just go and check where our data is stored without any clashing and confusion. So you can store the project where you can put the data, that is all the FASTQ files, in one particular folder. And then what you can do is you can create a section called as result section. That is, whatever files you get generated after running the KIME commands will be stored in the result section, and just keep in the main directory metadata and manifest.csv. The CSV is a comma separated file and TSV is a tab separated file. You can generate a metadata.tsv file as well. That's not a problem here. So after generating this, so in this manifest, you will have the path for metadata, and you will have the path for all the data files as well. And whatever you create, and you can even put the result path also, so that the results get stored. So this and all we'll be doing when we start running our command. So I will suggest you guys to have this sort of a directory structure, so that you will be also able to understand and work in a systematic order. So now when we run KIME, there can be certain errors. So I have already mentioned here some, and these are a very common errors that might happen when you are initially a beginner and starting to learn KIME. So first is incorrect file paths. So what happened with me is when I learned KIME and I gave wrong paths in my manifest.tsv. I thought that we just have to give paths, but I gave a slightly wrong path. So my none of my commands worked, because the path was itself path was itself wrong. So my command is trying to access manifest.tsv. In that, I have written the path so that using that path, it can go to my input files. But that path itself is wrong. So my command was never able to access the input files, and therefore I didn't get a result. Second is the data format. So the way we put the data in the manifest, that can be a little problematic in a wrong format that we need to cross check. Then missing metadata. Metadata might, whatever we are trying to look for in the metadata might be missing. So that can also lead to certain failure of the KIME2 pipelines. Then environment not activated. Then the KIME command only won't be found. And then typing errors in command, that is very common and most common, that we guys need to look into more consideration, because always typo errors happen. And then we tend to figure this out at the very later parts, when the entire pipeline is done and we are already exhausted there. What is happening? Why is it not happening? And then we just look into the command and see, oh, we just wrote I instead of A. And that's a very lame error that happens and at the end, but it's still a very important error that happens. So that's what are the best practices that you can follow so that we can avoid any error that can happen in KIME. First is always activate the environment. Before running any command, always activate the environment where you have to work. So after that, you will not get any error regarding the environment, that the command is not found or whatever tool you are using is not found. Then use an absolute file path. So you should know whatever or wherever the file is stored, or either open the KIME in the same directory as your stored file, so that you don't have to put in a lot of paths. But if you open it in another directory, then you make sure that the file paths are put properly. Then before inputting any file, you need to first open the files, that is the .tsv, manifest, metadata, fastqc. Just open it once and check if those files are valid. Because if those are not validated and if they might have some error, then the later analysis will also cause a very much problems. And we need to keep backup of data. So whatever data you are using, you need to keep backing up and never work on the raw data itself. See, if never modify the raw data. If you have a raw data fastqc files, don't modify that directly, because then you might lose the raw data, and if your result doesn't come, then you don't have the raw data back to analyze. So always make the analysis and results of that raw data, but never modify it. and then document all the commands that you have done. Make a separate folder or make a separate text file where you document each and every command that you have wrote, and even if you wrote, and even if you can give a little explanation also for that command, like say you use that command to do demultiplexing. So you can write heading as demultiplexing command and then write the command. It is very easier to later come and analyze and understand what did you do, and if there's some error that happens, then you can also check. This is just a manual way of documenting. Anyways, you know that provenance tracking is already there, but this becomes a little more easy because you can easily understand this because you have written yourself. So this becomes a little more beneficial, and this may help you also to record whatever you have done and at what step you are and all of that. So this was all for today's session. We learned about KIND, what are the plugins and how to set up the environment and all of that, and what errors and mistakes not to do, and how does the command line basically looks like for this particular time analysis. So this was it for today. In the later sections, we'll be talking about more about metadata and all of that and how to start it. So I hope that today's session was clear, and I guess both of you understood. If you guys have any doubt, you can just unmute yourself and ask me your doubts.
Oh, ma'am, can you tell us what are all the common errors that you have gone through by using this KIND installation?
Sorry, your voice is a little low. Can you speak a little louder?
Can I hear now? You can
Yeah,
hear
I
me?
can hear. Yeah.
Ma'am, you said I'm asking you that what are all the common errors that you have gone through while installing the KIND tool?
In installing the KIND, you mean the Conda environment installation?
Yes, ma'am. Conda environment.
So when I installed the KIME, so initial error that I got was first I was trying to install KIME in my base environment, but it didn't happen because of the Python version clash, and because of that, I couldn't install my KIME in one go. And then later I kept on facing the same error. I had to even uninstall and reinstall my Python, but then I understood that I have to create an environment, and then I created a separate environment. I installed my Python version first, and then I installed KIME in that. Then that error got automatically it got corrected, and my KIME got installed.
Okay, ma'am.
Any other questions, guys?
No, ma'am.
No, ma'am.
Is today's session clear to both of you? Like, did you understand whatever I taught?
Yes, ma'am.
Okay. Just practice once. Just go and check whatever commands I have written in the presentation. I'll be sharing the presentation now on the drive. Just go read through it, check the commands. I think we have the next session after 2 days. So understand everything, how it is working, and then we'll move on to the next part of it. And if you have any doubts, you can always convey your doubts and just go through it. I will suggest just practice once, try creating the directories, try using those basic Linux commands that I have taught. Don't create the directories manually, use cd, use mkdir, whatever commands I have taught, so that you will get a hands-on practice as well. And then we can move on to the next later part where we will be just analyzing the commands. And you can just open the KIME help and KIME version and look into all the plugins that are present. And if you just want a little more introduction, you can just read about it a little. Even that mentioned in the presentation, you can just read a little more about so that you are a little more clear. And any doubts, you can always just convey them to me.
Yes, ma'am. Thank you.
Okay. Thank you, everyone.
Okay. Thank you, guys.